I'm so happy to be here. Thank you to the organizers for inviting me and putting together this exciting meeting. By training, I'm an ecologist and I mostly do field work and experimental work. But throughout my career, I've been very lucky to work closely with mathematicians and statisticians. And I feel like that interface where you're trying to ask challenging ecological questions and you're coupling that with method development in mathematics and statistics is really, to me, one of the most exciting areas of science. And so to me, this is what this meeting is all about and what the center here in Chicago is all about. So super enthusiastic to be here. And I put together a talk where I'm trying to go over some of the older studies also where we've done this method development coupled with very intensive field data collection. And indeed, I'm very interested in disease. I work on disease mostly in wild populations, and that means that these are these really messy systems. So it's a lot of noise that we're constantly trying to control so that we can extract the signals that we're interested in. And increasingly, we're interested in also pathogen diversity, whether it's strain diversity in a single host individual or a host population, or the entire microbial community and the pathogenic microbes in that community. And so for disease biologists, the very deceitfully simple question that we're trying to answer is that who becomes infected? I've been thinking about this question throughout my entire career, and I would say that after COVID, most of us are probably thinking about these questions almost on a daily basis, especially in situations like these. If someone comes with an infection, some others are likely to acquire it, and which ones are the ones that will acquire it is very interesting. Here I show a very simple schematic of the SIR model that identifies that you have an infectious individual surrounded by susceptible individuals. Through time, infection will spread, and some individuals will recover, while some will remain infected and will then transmit that infection to others. It could be a seemingly simple process, but it's complicated by a whole variety of factors that disease biologists are trying to disentangle from one another. In the 1960s, Stevens introduced what's called the disease triangle. And I've been thinking, I've probably never given a talk to any audience without introducing the disease triangle, because although it's simple, it very elegantly identifies the three factors that have to align for disease to develop. In all its simplicity, it states that you need to have a susceptible host, an infectious pathogen, and environmental conditions that are conducive to infection development for disease to develop. But if you start thinking about the levels of complexity at the corners of each of these disease triangles, you realize that it's a much more complicated issue. You have different host species that might differ in their susceptibility, host individuals, genotypes, host condition, developmental stage. The same applies for pathogens. Pathogen species and strains will very much differ in their ability to cause infection. And the environment, whether it's an abiotic environment or biotic environment like the microbiome, is going to have a major impact on who becomes infected and who doesn't. And so to me, if I would have to say the three main classes that generate analytical challenges in understanding disease biology, I would say that it's really trying to tease apart the key elements of infection in the disease triangle, the hierarchical nature of infection dynamics, and the spatiotemporal nature of disease data. And so this is just to illustrate the kinds of complications we have even within the disease triangle. So you have your host species or host genotype, you have your pathogen genotype, but any infection that we observe is going to be an interaction between those two. And we would like to be able to say to what extent it's been determined by the host genotype and to what degree the genotype of the pathogen. And that can be really hard to do because it's always a component of the two. We can have direct impacts of the environment. We know, for example, for a lot of fungal disease that are infecting agricultural crops, they tend to like humid conditions, they tend to like relatively warm conditions. But at the same time, it's not so straightforward. We have some host individuals that respond differently in their susceptibility, depending on the abiotic conditions. We can have differences among pathogen species or genotypes in how they respond to abiotic variation, and that interaction between the host and the pathogen can vary according to environmental conditions. And the microbiome that we're increasingly starting to understand and tease apart its importance brings a whole other level of complexity. So you end up with these pathogen-pathogen interactions, pathogen-pathogen host interaction, pathogen-pathogen host environment interaction. It goes on and on and on if you take into account that a single host individual can be simultaneously exploited by even some hundreds of microbes at the same time. So it's not an easy task. The hierarchical nature of infection dynamics, and by that I mean that infection is happening at multiple levels at the same time. At the smallest unit, you can have the host cell that can be manipulated and exploited by a single microbial organism or by multiple different microbial organisms. And here I've shown as an example fungi, bacteria, and viruses infecting the same host. And that's a very likely scenario for hosts occurring in the wild. Then we have the host individuals, and they can be exploited by single pathogenic microbes, or they can be what we call a co-infection scenario where you have different microbes infecting the host at the same time. And depending on the infection status of the host, of whether it's uninfected, infected by a single pathogen, or infected by multiple pathogens, is going to have an impact both on its probability of becoming infected, but also on its probability of transmitting infection to other host individuals. And then finally, we have hosts occur in populations. Transmission of disease is typically distance dependent, and so how pathogens move between these host populations is a critical component of disease dynamics, and so how these are spread out in space is going to be very important. And finally, no host in nature occurs as a single entity separated from other species, but rather host species are embedded in communities that will vary in how diverse those communities are. And we're starting to learn quite a bit about how that diversity of the surrounding community is impacting disease dynamics. We know that biodiversity can have the ability to dilute disease transmission. And so that's one of the benefits of biodiversity that we're currently looking into in my group. And of course, space plays a major role in disease dynamics, like in most other biological questions. Imagine that you're a pathogen infecting a host in this setting. Your ability to move to the surrounding host individuals is going to be much easier than if you're a pathogen infecting a host here, because your transmission path is going to be interrupted by these gaps where there are no hosts available. And so the degree of fragmentation is going to be one of the key determinants of how infection spreads through space. And the more spatial complexity a biological system has, the more also other habitat complexity it's likely to support. And like the disease triangle identified, we know that varying environmental conditions can also have major impacts in disease. And given that transmission is a distance-dependent process, bringing together the distances separating host populations and the quality of these host populations is the final key determinant, so that essentially spatial structure is going to be one of the key determinants for disease dynamics. And so today I'll try to tell you something about what we've learned about the determinants of infection in the wild and how we've tackled some of the analytical and experimental challenges that come with these noisy data. I have to say, plenty of challenges remain, and we have heaps of data. So if these are the kinds of questions that you're interested in exploring, please come and talk to me. I would love to talk with you about ideas on what could be done. Much of my work has been done with the host plant Plantago lanceolata. It's native to Europe and Asia, but it's an invasive species here in the US, but it's a very commonly found species. It's not an impressive looking species. Always when I talk at agricultural conferences, they have a hard time understanding why someone would study this weed when you could be addressing real issues like diseases of crops. It's naturally infected by a powdery mildew fungus that you can see as these white lesions on the leaves, Podosphera plantaginis. And this is one of the main advantages of this system, that you can see infection with the plain eye. in the field because it allows you to do field studies without any serological tests or PCR tests. You can just go into the field and see which plants are infected and which aren't. But there's another reason why I've been working on this particular system, and it links to the work of late Professor Ilkka Hanski. He was a population ecologist, and throughout his career, he was interested in multiple questions, but perhaps is best known for developing the metapopulation theory that I will explain to you a little bit later. But essentially, it's trying to understand species persistence in fragmented landscapes. And he decided to study this question using the Glanville fritillary butterfly, and Plantago is one of the two host plants of this butterfly. And in Finland, they don't occur on the mainland, so they only occur on the archipelago. And Ilkka decided that he's going to study the dynamics of this butterfly in the archipelago that falls between Finland and Sweden. It's an ideal setting for looking at spatial questions because the archipelago in itself imposes this very pronounced spatial structure on the system. The larvae of the butterfly weave this nest on the host plants, and so it's easy to see in the autumn. You can simply go to these meadows and you can look at whether you find a butterfly nest or not. And he started these studies in the early 90s, and he decided that he's going to map all Plantago meadows in Åland. And I'm sure at the time everyone thought he was crazy, but he was a very determined, ambitious man. And so today we have a database of about 4,000 meadows that we visit on an annual basis. So we have about 40 undergraduate students who go to the archipelago for about 2 or 2.5 weeks, and they go through each of these meadows and they check them for the presence of these butterfly nests. But since 2001, when I started my PhD, they also started looking for infection on Plantago, so this powdery mildew fungus. And we've managed to keep this long-term study going on ever since, and it turns out that actually today it's the largest epidemiological data set collected from the wild. And indeed, like I said, one of the benefits is that you can see infection readily with the eye, but there's of course many other questions that we can't answer with that field data alone. So we have been developing molecular tools. So we've sequenced the genome of both the host plant and the pathogen, so we can really start to get at the levels of diversity that the system has. And we have the epidemiological data. And so coupling the genomics data with the epidemiological data really offers opportunities for trying to understand spatiotemporal disease dynamics and how diversity in a natural pathosystem is distributed. And there are some other experimental opportunities that the system offers. There are no ethical constraints. That's one of the main features of disease biology. In my case, we can, and I have killed thousands and thousands of Plantagos, and no one cares. It's also a specialist pathogen, so we can do field transplant experiments, and we don't have to worry about contaminating fields of nearby farms. The pathogen can be purified, so we can simply collect an infected leaf. We can isolate some of the spores and transfer them to a new leaf. And once we repeat that step multiple times, we think that we're dealing with pure strains. The downside of the system is that it's an obligate pathogen, and so we can only maintain it on live leaves. And so having a big strain collection is very time-consuming and is also just maybe a bit at risk of contamination. But also both the host and the pathogen can be cloned, which is also a huge advantage when you're trying to understand the impact of genotype on the infection dynamics. And we can also survey the life cycle of the pathogen without having to destroy that infection. So it starts out by germinating on the host surface. It produces this mycelial mat, and it then starts to sporulate. The spores of powdery mildews are produced in chains. They just rise up from the surface of the host leaf. There can be up to 30 spores on a single chain. It's actually very beautiful. If you look at it under the microscope, there's a tiny drop of water in each of these spores that will help it sporulate. And so in the world of spores, these are quite big. They don't travel very long distances. And then finally, I did my PhD in a lab where others worked with butterflies and flying squirrels and bears. And so my powdery mildew was never particularly exciting, for example, for student projects. But one of the nice things about it is that, like Stan Harper said in the 90s, plants then still to be counted. So you can mark individuals and populations. This is a perennial plant, and you can revisit that over and over again without having to chase it down. So collecting spatial data is pretty amenable. This is what a classic infection matrix looks like for this interaction, and it's not like one that would be similar in many others. Here I have 8 host genotypes, 8 pathogen genotypes. 1 depicts an infection and 0 depicts a resistant response. And you can see that the same host genotype is going to be susceptible to some strains of the pathogen while resistant to others. We have a few host genotypes that are susceptible to everything. Overall, susceptibility is more common as a trait than resistance. From the side of the pathogen, we typically have those that are capable of infecting some host genotypes while not others, while we do have a few strains that are capable of infecting all the host genotypes that we test them on. And that's in itself, I think, a super interesting question, that how do you get co-occurrence of strains that really vary in their ability to infect the hosts? This is what the disease triangle and all its complicated interactions looks like in reality. So we have these 8 host genotypes, and we tested them with the 8 pathogen strains under different nutrient treatments and under different temperature conditions. And we see that if you look at their different life history stages, like germination time or spore production time, is that they respond in a very genotype-specific way to the environmental gradient. So there's no host genotype that outperforms all others across all environmental gradients, but rather they switch their ranks. And I think that's one of the key mechanisms through which diversity of these strains and hosts is maintained in this system. And the same is true if we look at spore production rates of both the pathogen and the host. So genotype environment interactions are very important. So this is what the archipelago looks in reality. We don't, in fact, go to the smaller islands. It would be logistically very challenging. We do most of our work on the main island. It's about 50 by 70 kilometers. Much of it is naturally fragmented. This host plant occurs on very dry meadows, but of course also human intervention of agriculture and other developments are breaking up the meadows and creating this highly fragmented structure. The metapopulation theory that Ilkka Hanski was developing really views at persistence at the metapopulation level, where you're not thinking about birth and death of individuals, but you're looking at colonizations and extinctions of local populations and trying to predict whether a species can persist at the landscape level in a balance between colonizations and extinction. So these are really the demographic events. And the genetic landscape is modified not only by the extinctions and colonizations, but by gene flow that links the local populations together. And the size of the local patch, which is metapopulation terminology for local population, should be the key determinant of which populations are colonized and which are not. And it's a really intuitive framework for host pathogen interactions, because there it's pretty intuitive to identify what's a suitable patch, because that will be where you have your host individuals. This is what the Plantago meadow network looks like in Åland. Plantago can be found everywhere. There's about 4,000 local populations, but even so, there's also some structure to it. So the density of the network tends to be higher here and lower in the central parts of the island. I didn't put the equation for connectivity because usually I try to hide all the math in my talk, whereas I think that's the part that you would be more interested in. But we calculate connectivity, which essentially for each of these populations sums the Euclidean distances separating these populations and also takes into account their area and weighs it by a dispersal function of the species of interest. And with that, we can see that we have a range of connectivity. So these are what we consider to be the highly connected populations, where we think there's a lot of gene flow among these populations. These are the intermediately connected, and these are the isolated populations, where we assume less gene flow arriving to these populations. And so, of course, if we're trying to understand how the pathogen is moving across this spatially structured network, accounting for spatial structure is very important. And there's, of course, a multitude of ways to quantify and control for spatial autocorrelation in your data. The method that we've been using the most is what's called INLA, the Integrated Nested Laplace Approximation. It's a very broad category for fitting Bayesian latent Gaussian models with varying temporal and spatial structures. One of the benefits of INLA is that it should be relatively fast to converge, but I have to say with about 30 years of epidemiological data each year from thousands of populations and tens of covariates, fast isn't necessarily the word I would classify or describe running these models, but nonetheless, has been a very useful tool for our purposes, and it really allows you to quantify the shape of both spatial and temporal autocorrelation rather than making a priori assumptions about it. And so this is what the disease dynamics looks like. It's an older analysis. We actually haven't done a simple temporal analysis for a long time, and we definitely should. In metapopulation analysis, what we're interested in, which host populations are occupied, which ones go extinct, which ones will be colonized, and which ones will persist year after year. And here I'm showing you one snapshot of what these metapopulation events look like. The blue squares are the populations that have persisted. Red triangles are populations from which the pathogen has gone extinct, and the yellow dots are populations that the pathogen has colonized. And you can see that there are not that many blue squares. So it's a highly dynamic system that really seems to be operating in a very classic metapopulation way of when populations go extinct, and we think this mostly happens during winter, then new ones will be colonized during the summer epidemic season. An interesting feature of this system is that overall, infection levels at the metapopulation level are pretty low. So typically less than 20% of the host populations are infected, which is, I think, again, interesting in itself. Much of that has to do with the very high extinction rate of the local populations and the fact that colonizations, although they are important and successful, the season in Finland for the pathogen to do so isn't so long. And so once it really gets going, temperatures start falling down and the epidemic season stops. And so that means in practical terms that the years that the pathogen has managed to persist in any given host population, at this time about half of the host populations had never been infected by the pathogen, and only a handful had been infected by the pathogen for the whole duration of these studies, which was about, I think, 14 years at the time. We find that one of the biggest determinants of which population becomes infected is the size of the local host population, which makes perfect sense. It's simply a bigger area to capture arriving spore. The spores are dispersed by wind, and so it's simply a game of probabilities. Distance is a key player, so that as the distance from the nearest persisting pathogen population increases, the probability of that population to become colonized by the pathogen decreases. We also know that the landscape structure plays a major role. We find consistently that if the local meadow is by a road, it has a much higher probability of becoming infected than those that are not roadside populations. We actually have a beautiful model on how the road network works. I didn't include it in this talk, but please come and talk to me if you're interested in kind of roadside ecology. But in our case, much of it has to do with the fact that although we have this mapped population network, Planteco grows along the roadsides as well. And so I think these serve as dispersal corridors into the local populations. And also the airflows created by cars can enhance transmission of this pathogen. We also have some signals that abiotic conditions matter for the pathogen. So here we were looking at August rainfall, and we found that unoccupied populations tended to experience slightly higher rainfall. And rain in this case, all fungi benefit from rain to some degree, I would say, but it can also wash away spores from the leaves as infection hasn't started to develop yet. And in that case, it can have negative impacts on disease dynamics. Okay, so what about host resistance? We have this super complicated system of pronounced spatial structure. And is there variation in host resistance, and does that matter for infection dynamics at such a large scale? Overall, what's good to keep in mind is that in natural systems, variation in resistance can be found at any level where you look at it. If you look among species, there will be tremendous variation in how much species invest into resistances. In a recent study, we used published genomes of plant species, and we annotated resistance genes from those genomes. And we found that there's variation from having about 30 resistance genes to having some 2,000 resistance genes. And that in itself can be considered quite a difference in investment towards disease resistance. And not a part of this talk, but I'll just quickly show you. In wild plants, you have the highest levels of resistance investment, whereas in agricultural species, it's significantly lower, suggesting that something has purged excessive resistance from crop species that have undergone breeding. We also find variation in resistance if we look at within species. Here we quantified variation both in pathogenicity and resistance across different wild host pathogen interactions, and we looked how much of that variation could be found within populations and how much of it could be found between populations. And you can see that in some systems, most of the variation is in fact even within populations. So although resistance should be such an obvious trait to invest in, why not be resistance? Because it can help protect you from infection that could ultimately kill you. It's one of the most variable traits that we have in nature. The classic explanation for how this level of variation is maintained is the growth defense trade-off. That suggests that any investment you put into defenses will come at the cost of lower growth. It's an extensively studied hypothesis with very mixed results. I think one of the challenges in studying this question is that it's so hard to define a full defense phenotype. Like, how do you challenge a host individual against all the things that it will be eaten by or infected with? And so we also haven't done that, but we decided to focus on these resistance genes. They're not the only resistance mechanisms that plants have, but they are a major component of resistance. And so we compared, sorry, I'm lacking a label here, but we compared the proportion of the genome that's invested into resistance genes against the growth traits of that plant. And we find that in wild species, there's a significant negative association between investment in defenses and ability to grow. And we don't find that same negative relationship in agricultural species. So again, suggesting that this natural mechanism of maintaining variation has been lost through breeding. In Plantago, there's a lot of variation in resistance, both within and between populations. This was one of the first studies I did in my PhD. I sampled host individuals along about a 10-kilometer scale, and I tested their resistance against 4 different pathogen strains. And half of these populations were infected in the field and half were empty. And I found that healthy populations, in fact, had higher levels of resistance than infected populations. And within each of these populations, how the plants responded and were infected by the 4 different pathogen strains varied quite a bit. So variation both among populations and within populations to pathogens. And the unexpected feature of the earlier study that I showed you of the disease dynamics in Åland was that we found that host connectivity had a negative impact on disease dynamics. Metapopulation theory would predict that as you have connected host populations, their probability of becoming infected should be higher, simply because it's easier for the pathogen to move around in that connected host network. And we found the opposite of this. So we found that the pathogen was more likely to go extinct from host populations that were highly connected. And and we also found that when we looked at empirical data through inoculations, we found that as host population connectivity increases, so does host population resistance. And so it seems that there's enough gene flow in these well-connected host populations that could maintain sufficient levels of diversity that could provide protection against the pathogen. And we did a study. This was led by Leila Höckerstedt, who was a PhD student at the time. She sampled 20 Plantago populations from across the archipelago. Some had disease history and some hadn't. And we had classified them as being either highly connected populations or isolated populations. And for an inoculation study, we infected them with 4 pathogen strains so that each host individual could acquire a phenotype where it would either be susceptible to all of the resistant to all of the pathogen strains, or no, this is resistance. Yes. So either resistant or susceptible to all of the pathogen strains. And I don't think the colors aren't showing up very well, but the greens indicate connected populations. And we find that the connected populations really tend to cluster in this part of the matrix where you have, first of all, quite a bit of variation, but also it tends to cluster in this more resistant part of the matrix, whereas the isolated populations tend to cluster here very much in the being more susceptible. So really providing support for the earlier study that we had seen in the epidemiological analysis of these data. And if you just average these of looking at, for example, Shannon's index of diversity, we find that indeed the connected populations have significantly higher levels of diversity and also average level of resistance than the isolated populations. We didn't see significant impacts of disease history on levels of diversity, which you would expect based on coevolutionary theory. And it could be that in a natural system such as this one, where there's ongoing gene flow and also mortality due to a wide range of other things, it's not showing up in a way that we would expect it to. We were also interested in whether we can see any impact on host population fitness by the pathogen. I mean, this is by definition, pathogens should have a negative impact on their host. But if you look at examples of this from field studies, there are remarkably few. And I think one of the reasons is that we have so few comparisons of really tracking populations with and without infection. As disease biologists, we tend to put all of our focus in situations where there is infection, that we tend to ignore the uninfection scenarios. But of course, our epidemiological data keeps track of both of those, so it really helps. And so we were looking at changes in host population size. And again, we were interested in whether that differs between the connected and isolated populations. And we find that when we compare populations with and without the pathogen, we find that always with the pathogen, population growth rates are lower. And this is true for the isolated, intermediate connectivity and high connectivity populations. But the difference is the greatest in the isolated populations, so that if the pathogen succeeds in getting to these isolated populations, it will do the most damage, because these are the populations that contain the least variation in resistance. And I have to shout out to Elina Numminen. She's a mathematician who was a postdoc in my group for several years, and she was responsible for carrying out these analyses. So the take home of this part of the talk is that both inter- and intraspecific diversity in resistance are found everywhere. In the Plantago system, we see that it's spatially structured so that resistance diversity really clusters in those well-connected populations, and we find less diversity in isolated populations. And although it's a messy system, we still see that the spatial structure is so important that it can also mitigate the impacts that disease has on the host populations. Okay, so what about this diversity of pathogens that I mentioned at the beginning of my talk? The powdery mildew certainly isn't the only pathogen infecting Plantago in Åland. For years, I kept seeing these yellowish lesions, which according to literature could be an indication of viral infection. But viruses are a nightmare to study in the field because they typically don't cause very conspicuous lesions. So you have to go through a whole range of testing to really know what's there. We developed qPCR primers for 5 viruses that are quite commonly found infecting Plantago in Åland. And in this study that was led by Suvi Sallinen, who was a PhD student in the lab at the time, we knew nothing about interactions between Plantago and these viruses. And so the basic question we had was that, do these Plantago genotypes differ in the kinds of viral communities that they acquire? So we selected 4 genotypes. We had no a priori knowledge about their virus resistance. We cloned them into tens of individuals that we then transplanted into 4 populations across the archipelago, so that each genotype was represented, I think, by 80 replicates in each of these populations. We left the plants there throughout the epidemic season that for the viruses is from beginning of June until August. We then collected samples, and through qPCR primers, we identified which viruses were infecting the host plants. And here, the color codes show you that we have both single infections by the 5 different viruses. We have infections that are caused by 2 different viruses, by 3 different viruses, by 4 different viruses, and even all 5 viruses could be present in a single host plant. And the data here are organized according to the genotype and by the populations that they were transplanted in. And you can see that there's quite a bit of variation. So the genotype 609 seems to acquire the highest viral infection levels and also highest viral diversity, regardless of where it's being transplanted, whereas then genotype 28186 seems to be having the lowest levels of infection and diversity, and then everything in between. And to analyze these data, we took what's called the HMSC approach, hierarchical modeling of species communities. And it's a very intuitive model to use when you're trying to understand distributions of multiple species at the same time, because it allows you to analyze the joint responses of those species to the covariates that you have in your model. And so we're interested in either the occurrences in our case, but you could also be looking at abundances of these species. You can include your covariates into these. You can also account for the phylogeny, because we assume that the closely related species may be more likely to respond in similar ways to environmental variation. You can also include traits in your model, because some traits might also explain some of that variation. But at the end of it, in addition to allowing you to quantify the importance of the different covariates in your model, it also gives you residuals that you can use to estimate whether, after accounting for all the other factors, you have signals that species are occurring more or less than you would expect by chance with each other. And this is, of course, one of the most fundamental questions that ecologists are interested in, that is there indication of species interactions? Is there indication of antagonistic interactions where they would occur less than you would expect, or indication of more, which could be indicative of some sort of mutualistic association between these species? Although a word of warning, reviewer 3 will immediately tell you that it could be simply due to an unmeasured covariate, and that's absolutely true. So we should treat these more as hypotheses that can be tested experimentally. But it's a very useful class of models developed by Otso Ovaskainen, who's a close colleague of mine. So. And for our data, we were interested, of course, in the corners of the disease triangle. We were interested in attributes of the host, attributes of the pathogen, and attributes of the environment in how they shape these within host viral communities. And we discovered that the host is by far the most important component, understanding levels of viral diversity. So we took into account the host plant genotype, which was the most important one, with explaining 42% of variation in this data, the size of the host plant, and also whether it displayed signs of herbivory, which can have some impact on infection probability, explain some variation. The local population context, which we, in this case, we didn't measure. So it can be anything from temperature to precipitation, landscape structure, or vectors. These viruses, we assume, are being transmitted by vectors from one plant to another. Explain, and I was expecting it to explain more, but it explained 29% of variation in this data. And after controlling for all of that, we found that about 14% of the data was explained by associations between the viruses, and these tend to be highly positive. We also find that the viruses responded differently. So, for example, Capula virus, for it, host attributes were by far the most important, whereas then beta partitivvirus environmental context seemed to be more important than the host attributes. So we get a nice sense at the community level, but we also get a sense of how the different viruses respond to the system. Well, how important is the landscape structure for virus diversity? Unlike the fungal pathogen that I talked to you about earlier, which is a specialist on Plantago, we have no expectation that viruses would have similar degrees of specificity. In fact, I would assume that many of them are also capable of infecting crops. And as we know, agriculture has replaced natural habitats across the world. And so we're having these situations where natural populations and agricultural fields are meeting each other, and there will be spillover effects both from the fields to nature and vice versa. And what are its impacts on disease dynamics? In this study that was led by Hanna Susi, she sampled populations from Åland that were either immediately adjacent to agricultural fields or those that were not. And she again was investigating these 5 viruses for which we have primers. And overall, we find that in these agricultural edge populations, we have higher virus infection prevalence than we have in the natural populations. And we also find that there's higher viral diversity in these agricultural edge populations than in the natural population, suggesting that there most likely is some spillover from the field to the wild. The dilution hypothesis predicts that the more diverse host species diversity is, the less infection you're likely to find. And we find that overall, we find some evidence for this. But once we start looking separately at the host community diversity and virus diversity, we see that the dilution effect only operates in the natural populations, whereas in those populations adjacent to the agricultural fields, in fact, it's reversed. So it seems that the influence of agriculture can also fundamentally change the basic mechanisms through which disease is regulated in natural communities. And as my final example, I'll show you what the full viral communities in Åland look like. So here we sequence the entire virus community from each of the host plants rather than focusing on the 5 for which we had primers. For this study, we selected 20 host populations from across the archipelago, and we found altogether about 40 different viruses. For analytical purposes, we had to leave out the rarest ones, but nonetheless ended up with a very diverse virus community. And there's a lot of variation again. So for example, if we look at population 3222, we find that about half of the host individuals that we sampled were not infected by any virus. The rest were infected, but typically only by 1 or 2 viruses. In the extreme scenario, we have host population 861, where only one of the individuals didn't have any infection. Most hosts were infected at least by 2 or 3, and one was infected simultaneously by 24 different viruses. So that's tremendous viral diversity in a natural setting. And of course, we don't really know anything about these viruses, what their ecological roles are. And the colors of these pies indicate the levels of complexity of infection. So the whites are 0 infection, and the darker the color, the more complex viral community you have within that host individual. And you can see that the full picture is highly diverse. And here we took what's called the conditional random fields approach. So again, it's analyzing joint distributions of species. But one of the benefits of this analytical approach compared to the HMSC approach that we use quite often is that it also allows you to identify. So you have your covariates in your model. And here we had the host individual, its size, whether it has signs of herbivory. We have the connectivity of the local population. We have the size of the local population. We have the plant diversity of the local population. We're looking whether it's close to agricultural fields. We're looking at abiotic factors. And we're very interested in this, whether we find associations between 2 different viruses. But in addition to that, we're really interested in whether we can find these higher order influences. So whether we see virus associations emerging in response to a particular covariate or in response to a particular virus. And for this purpose, this CRF approach is highly useful. And we found that in our model, we found the most support for explaining viral diversity, the agricultural fields again, even when looking at the full virus diversity, signs of herbivory on the plants, so suggesting that really maybe the vectors have easier access to plants once they've received some damage. But we also see an impact on the size of the host population and plant diversity of the host population. We find that the virus communities seem to be highly connected, and we only see evidence of positive associations between these viruses. So the wider the path between two viruses, the stronger the support there is for this path. But so you have a lot of connections and pretty high support for each of these. If we filter, and here is one really nice example. So that this approach allows us to do is that, for example, we find this association emerging between Rhopaviridae and Betaflex viridae arising only in populations that are adjacent to agricultural fields. So really, a really cool example of the complexity by which these viral communities can be structured. If we filter this network through the covariates that we included in our model, we find that, and so essentially removing all of those associations that could be simply due to shared similar responses to the covariates, we still end up with a nice network suggesting viral associations, some of them quite weak, whereas some of them really quite strong and could be very interesting to investigate any biotic interactions in more detail. But with that, the take home from this part of the talk is that human land use has major implications both through overall fragmented structure and what that landscape consists of, whether it's agricultural land use or wild. And our data suggests that biotic interactions are the key determinants of disease dynamics in the wild. We find significant impact of host genotype and host population across all our studies on the different pathogens that we've looked at. And we also see strong evidence, at least from this field sampling approach, of also pathogen-pathogen associations determining the complexity of natural pathosystems. With that, I want to thank my amazing lab members who are a joy to work with, and also all the alumni whose work I talked about here. I want to thank all our funders. I want to thank the students who go to Åland at the end of each summer and miss a few lectures, which other professors always complain to us about. And I want to thank you for your attention. Thanks.
Yeah, thank you. That was awesome. I was curious about this idea of weather and wind. And I was curious if you can measure wind locally and use that as a way to predict what's going to happen in the future. And then secondly, just on like stochastic weather events, like is a single gust of wind sufficient to change like long-term behaviors?
Yeah, that's a very good question. So we've included in our models just latitude and longitude, and we see some signal of that even after controlling for other spatial structures. And the prevailing wind condition in Åland is from southwest, which could be an indication of that it's then determining kind of infection patterns towards the east. And we've done some wind tunnel experiments. So these are operating at a very different scale. So we're really looking at centimeters. So we were interested in quantifying the kernel of local dispersal, where we artificially created these wind bursts, and we do see an effect on it, but doing it at the Åland scale would be harder. I mean, I'm sure there's ways to do it, but we haven't looked in more detail into it. But definitely, it's important because it's the main mechanism for long-distance dispersal for this fungus.
Thank you. That was extremely interesting. It seems that in the graph, when you look at the relationships between different viruses, there is some implied positive relationships between them. And I'm a biologist, so from a biological point of view, you had two viruses in the same cell. They technically are going to compete for the resources in that cell in order to reproduce themselves. So how would one explain that two viruses are cooperating?
Yeah. So, well, first of all, so the way we identify viruses in this study is that we actually sequence the plant's small RNAs, which is essentially like a resistance response to those viruses, which is nice in the sense that we know that it's an active interaction with the viruses rather than like bacteria that are just laying on the surface of the plant. But even so, many of these viruses could be in a fairly latent state in the host where they're not necessarily replicating. Like most of these plants that we sample in the field, they don't show clear symptoms of viral infection. So we don't assume that they're necessarily infecting the plant actively at that time. Even if they were, I could imagine a few different reasons. So first of all, simply it seems that host genotypes, so that we do have these host genotypes that just tend to be more susceptible to viruses than others, which could allow them to acquire viral diversity. Vectors are the key players here. So if vectors have some preferences towards infected plants, for example, they could pick up different viruses and bring them to the same host. And there again, then this is the disease triangle. So you could have something in the environment or in the density of the hosts in certain parts of the population that then makes infection aggregate.
Thank you so much. So you talked about in the example with the fungal infection in Plantago, you were able to quantify a number of resistance genes. How do you quantify a resistance gene? Because you could imagine something that has some completely external purpose that inadvertently happens to help with resistance.
Yeah, so we use a very widely accepted bioinformatics pipeline that's looking for leucine-rich repeats in the genes, and that's essentially a signature of a resistance gene.
Hi, nice talk, Anna. So in the spatiotemporal dynamics, when you were saying about the resistance, so does it also happen on different stages? Like in the first stage, if some isolated plants who are infected but try to develop some immune system. So in stage 2, those isolated plants can be completely immune, although they are isolated, because they have some defense mechanism that they have developed in stage 1.
So your question is that whether the isolated plants, if they become infected,
in different stages, in stage 1, once they are infected but they survived and they develop some resistance and defense system, then in different stages they are although isolated but completely immune from the viruses.
Yeah, that's a tough question. I mean, they're isolated, and so maybe they're more naive as a host population with less exposure to the pathogen than some. But of course, once the pathogen gets there, they seem to have fewer coping mechanisms. So, I mean, in principle, I assume that the process is the same in both. It's more a feature of the population level diversity that's diluting infection dynamics rather than something different in the infection stages.
Okay, I'm going to take one last question.
Hello, thank you for your talk and thank you for including the photos. I thought that was really nice that we can see who was part of the projects. When you talked about the populations that are close to the agricultural, I think you called them edge populations. It seems to me to imply that, for example, the pesticides or the genetic modification that we're doing to the crops might be negatively affecting the wild populations. And I wonder if that has directly been studied, even if the funders, depending who they are, may or may not like that. I think it's an important question.
Yeah, definitely. So we haven't looked in much detail into that, but there's definitely a lot of research looking at the effect of nutrients and pesticides and fungicides leaking into surrounding natural populations. Here we only tested for the soil nutrient profiles, and we find that it is different in these agricultural edge populations, which in itself could also change the infection dynamics. So for example, if you have higher nitrogen content in these populations, it could, through the host plant, then have an impact on the pathogen.